Hi everyone, and welcome to this interesting discussion of microbiology image based questions. I will be discussing 30 image based questions in this module or discussion. Okay, first is schistosoma X. So the first image you see is having a terminal spine, okay, and it is schistosoma hematobium. It resides in the vesicle and the pelvic plexus. The other image has a lateral spine which is seen in schistosoma mansoni, which resides in inferior mesenteric vein, and the veins which drain the inferior mesenteric vein. And the third image is having a rudimentary spine. It is schistosoma japonicum, and it respectively drains into the resides into the SMV. Okay. The next egg is ascaris. So ascaris have two eggs, a fertilized and a non-fertilized egg. So what you see in differences are fertilized egg has a mammillated coat. Other part is between the large ovum you have crycentric spaces, and unfertilized egg you have a thin coat and an atrophied ovum. Okay, next. So the next image is for Trichuris trichura. Okay, you see a barrel shaped egg with bipolar mucous plugs. This is characteristic of Trichuris trichura. Okay, next. So next what you see is you see the egg of hookworm. Okay, so the two characteristic points are you see blastomeres. Usually 4 blastomeres are present, and there is a clear space between the ovum and the eggshell. Okay, the other 2 points of hookworm are Chandler's index, which we calculate, which is the number of hookworm eggs upon the gram of stool, and the other point is cutaneous larva migrans is caused by the non-human hookworm species. Okay, next. Okay, this one is interesting. So enterobius, the egg is what you see is a plano-convex egg, and it has an embryoted ovum inside it. The other point is for the isolation of the enterobius we have two methods. One is a Scotch tape method. In Scotch tape method, as you can see, we take a tape, we the sticky end is placed between the thumb and the index finger, and you touch all the areas of the anal fold with the sticky end, and then you keep it on a petri dish and you deliver for the microbiological examination. And NIH swab is a similar thing where you have a cellophane at an end, cellophane paper at an end, and you collect the organisms after having a perianal swab. Okay, so enterobius, Scotch tape method, and NIH swab. Next, we have two larvae to differentiate between. One is a strongyloids larva and other one is a hookworm larva. So two important points are in strongyloids larva. First point is the buccal cavity which is there is a small thing. Okay, so like I can show you here. So this part which is there, you can see this part of the hookworm larva where you have a long buccal cavity, this much. Okay, almost 3 times of the buccal cavity. And the genital primordium which is there, it is more proximal in strongyloids and more prominent, whereas it is less prominent in hookworm. Okay, next. In protozoa, you see this is a characteristic image of clue cells. Clue cells are the organisms which are there, which are attached to the the vaginal epithelial cells in cases of bacterial vaginosis. And these clue cells are called as clue cells because we didn't know the actual organisms, and later it was discovered to be microorganisms which were Gardnerella, Mobilincus and all. Okay, and this is in bacterial vaginosis. Amsel's criteria used to diagnose bacterial vaginosis, where we need more than 20% of clue cells. The other point is Nugent score, which is on the microscopic examination, more than 7 Nugent score is diagnostic. Next protozoa is Giardia. Here you see a trophozoite of Giardia, where you see 2 nuclei. Okay, and you also see flagella here, and there is a parabasal plate. Okay, the other test for Giardia is an intro test. You have a duodenal cap here which is ingested by the person and the patient, and you attach the other part to the cheek of the patient. You get a duodenal sample, and then you examine it for trophozoites of Giardia. Okay. The next protozoa is Trichomonas vaginalis. Here similarly you see a trophozoite of Trichomonas, where you see there is an undulating membrane here which is not so clearly seen. We see flagella and you see nuclei. Okay, here you see strawberry cervix, which is characteristically seen in Trichomonas vaginalis. Okay. Next is Ballantidum coli, which is the largest protozoa, and it has a typical feathery appearance due to the presence of flagella. Okay, a large cell, typically a feathery appearance due to presence of flagella, multiple flagella. For fungus, the first image is for medullary body or copperpenny body, or these are seen in chromoblastomycosis. Okay, so this is a characteristic image. Next is sporotrichosis. In sporotrichosis, the image shown is an asteroid bodies. So sporotrichosis is a dimorphic fungi where you have the yeast form, which is cigar shaped, and a mold form which is floret shaped, and this is the asteroid bodies. Okay, the next image is a characteristic image of 4 systemic fungus. Okay, the first one is histoplasmosis. You see the histoplasma, you see the small cysts with narrow based budding. Okay, and these can be grouped within the macrophages. So the presence of narrow based budding is characteristic of histoplasmosis. On the counterpart, blastomycosis, as you see here, has broad based budding, and you see a figure of 8 appearance in blastomycosis. Okay, now the comparison between coccidiomycosis and paracoccidiomycosis. Coccidiomycosis, you see a typical cartwheel or a mariner's wheel appearance, and in coccidiomycosis you see a big what do you call it as multiple spirules in a big globi. Okay, next is the different microfilarias. Okay, so the first point what you see, so the microfilarias are differentiated between the presence of sheath and the nuclei at the tip. Okay, so for the first one which is there, you can see there is sheath for the microfilaria. The other point is that nuclei did not go to the tip of the tail. This is Wuchereria bancrofti. If you have two nuclei present at the tip of the base, tip of the tail, okay, that is Bruggeia malai. Okay, and if you have multiple nuclei at the tip of the tail, it is Loa, and Mansolina and Onchocerca do not have sheaths or nuclei at the tail tip. Okay, now few miscellaneous images. Okay, so this you see what here in the peripheral smear is a band form which is characteristically seen in Plasmodium malariae, and here you see a banana shaped gametocyte which is characteristically seen in Falciparum. Okay, Plasmodium falciparum. And the last image includes the peripheral smear which shows characteristic Maltese cross form of Babesiosis, which is also a protozoa. Okay, thank you very much.